Good evening, I guess. Usually it's good afternoon or good morning when I give these things. So this is an interesting subject that Bronwyn brought up, and so I went into it just sort of innocently. I know a good bit about it, but trying to put together a comprehensive overview of the subject is was a bit of a challenge. And the reason, and you'll see, the name of the talk is actually jelly fungi and similar cup fungi and slime molds. Because what it ended up really being, more or less, is exploring the odd excrescences that you find along the trail that are kind of amorphous and lacking any structure and really seem outside the bounds of definability according to any rules you might apply. And jelly fungi are, I think, notoriously, ill-defined both in appearance and in actually, when we look into it, you'll find that they're not very well defined in taxonomy either. Almost all mushroom guides will have a section called jelly fungi. As it turns out, like many fungi that look somewhat alike, most of them have very little to do with each other, except for this physiological aspect that they produce this gooey looking sort of semi-permanent that we give the name jelly from gelatinous because it sort of characterizes the texture. And you'll see four of them there in the picture, and we'll talk about each of those as we go through this. So the agenda, I always like to start with what we're going to do. To get sure we're on a baseline on what is a fungus and into the taxonomy, why you almost have to approach any discussion of a type of fungi through how it fits in with the entire kingdom fungi. And from there, and I'm going to use some of the introductory slides I use generally in my Master Naturalist, just for those of you who may not have gone through this before, it's a pretty good introduction to something that's pretty new in terms of science. Fungi were not a kingdom up until about 40 years ago, maybe 50 years ago now. So it is really a new science and still is in its very fledgling part. Then we'll go into what we generally group into the jelly fungi and what would constitute that group. And all guidebooks will have something called that because there's no other good place to put them. And we'll see some others that are put in the same category. So that's based on the third point, spore dispersal and fruiting body growth. That's really how things are divided up more or less within the current realm of guides, other than texts that go into structural details and atomic structure and things. Then we'll start looking at some specifics. The most famous of all the jelly fungi, the tree ear, and then we'll go into probably the most exotic, which is witch's butter. Those are two of the most very common ones also that you'll probably, if have not seen, you will at some point. And then we'll look at some of the others. And then toward the end, I'm going to talk about the cup fungi, why they're somewhat different, why they're somewhat similar. And they can be mistaken. There's a brown cup that looks a lot like the tree ear. In fact, it's in the book. It usually says it's a lookalike, so it's really the same, and you can't really tell the difference. Well, you can if you know what you're looking at, but that's true of everything. And finally, we'll end with slime molds, which is a really fascinating subject. And by the way, they're not even fungi, but they are also generally grouped with fungi, and people seem to be fascinated by them, particularly with the recent news about the very famous one that drew the Tokyo subway, which we'll talk about towards the end as a matter of rather interesting. So first of all, I always like to start, okay, what is a fungus? This is a definition. And as I mentioned to Bronwyn, most people, I got introduced almost all the time as the fungi, and still do. So I usually remind people, no, it's fungi. And you can pronounce it however you want, but the right way to pronounce it is fungi, as I show there. But the definition, this is the dictionary now. So if you've ever wondered what makes a fungus a fungus, it's those four specific points there: eukaryotic, heterotrophic, absorptive, reproduced by means of spores and branching, etc. So each of those terms has a particular meaning, and I'm going to just go down into one, make sure you understand what that means. So here they are in terms of the definition. Eukaryotic is having a nucleus. uh, everything except bacteria, basically. There's a slide on that following this. Heterotrophic means they get their food from other sources, as we do. Then absorptive has a star because that's what really distinguishes the fungi from the others in terms of being unique. Well, there are other things that are unique, but in the definition at least. And then finally, spores, which are not unique to fungi, of course. You have mosses and the whole group called cryptogamic plants, those plants where you can't see the seed, which has a long story all by itself involving Shakespeare and Falstaff, which I won't get into, about being hidden by them. So this is one of my favorite slides because it really puts the kingdom of life in a very visible format that I find quite appealing. When I first got into this, rediscovering life in about 1990 or 95, I thought it was, of course, most of us, absent any biological background, we just don't know of the plants and the animals. And this whole thing of prokaryotes and eukaryotes came up fairly recent, probably earlier than that, of course. But you'll notice on the left, you'll see blue, green algae, and brown algae, and they're all in different groups now. And so we've learned that algae is not a plant. In fact, it can be a protist and it can be a bacteria like blue-green algae. But the bottom, the bumper sticker at the bottom, and I'll use my pointer here. It doesn't come out very well. This is sort of my bottom line overcut at what life is in terms of the ecology of living things, or the ecology of biology. Plants produce, so they're allotrophic. Animals consume, they're heterotrophic, as are other things. But fungi reduce. So that's the cycle of producing hydrocarbons and then returning them to the atmosphere to be reused again. So it's called the carbon cycle and the nitrogen cycle. They're both related in recycling things that need to be in the atmosphere so we can use them to build life. So the second point, as you remember, was heterotrophic versus autotrophic. Fungi are somewhat more prolific in how they eat things. Most animals, have some sort of a mouth and a stomach and digest it, some internally, some externally, like spiders, but mostly we're used to everything being, sort of eating, I guess you'd call it saprophytic. So we mostly eat dead things. We can eat live things, but we don't usually. We usually kill it before we eat it. And plants are, I guess, in the middle. In terms of being alive or dead, you could eat both. But regardless, we're pretty much saprophytic. You'll notice that jelly fungi are in that list of saprophytic fungi, which is they live on things that are already died. I don't know of any parasitic jelly fungi, although I would rule out the fact there might be one. And there aren't very many parasites among the fungi. But this other one we're not going to talk at all about tonight is the most important, at least from the standpoint of the forest that is now the wood wide web, the mycorrhizal relationship between mushrooms and trees and plants in general. So that's heterotrophic. Spores, again, I point this out because if you go to an older field guide, spores were sort of the coin of the realm when you were trying to define where you were in the fungal kingdom, and they, even though you can't see them, they have microscopes now, and that's only from about the 17th century. And on the top left, you can see that was this is from an old mushroom book. These are some of the different shapes, and you'd go and look under a microscope, and that would define what you had. And the reason I'm harping on this a little bit is because spore formation and how you get the spores out there is really fundamental to how the fungal body, fruiting body, as we call it, as we'll go through briefly here shortly, is how it evolved. Probably no less exotic than the many tadpoles that are taken to make one frog adult, or the many seeds, or the blow and the anthers and the trees blowing their pollen all over the world, filling the air with it, so we all get asthma. So it's everything does this, but fungi are experts at it. And those are the spores, by the way, at the bottom right of that particular fungus, which you may know or remember as artist conch. And here are their colors, but completely worthless. And they don't do that anymore. They usually still talk about the spores' colors, but it's not of any particular value for field identification. In fact, it's of zero value for field, almost zero. Sometimes you can see the spores if they spread out. And finally, remember the other one, the star was absorptive. And this is really the important one from the ecology standpoint, because all the logs lying around would be still lying around if it weren't for the fact that they were decomposed. you may have seen this, the bottom right. You've probably seen that in the woods as you're walking about. What was obviously a former tree, which is what's left of it after the fungus has taken apart its cellulose. So it sort of falls into its constituent parts, and gradually then the bacteria will take over. The graph at the left shows how much of a underground testing of the in a kilogram of soil, how much of it is fungi among the non-plant materials. So it really is the way things work. So enough on that. So now we talk about what is a mushroom, and I use this as just a general term because everybody knows that and they think of that when they think of fungi. And really, all of the fungal world is fruiting bodies. And what I mean by that, we call the fruiting body of some fungi mushrooms because they look like a mushroom. Well, we call it that. And actually, I go through the whole definition of mushroom, where it comes from. It comes from the French word for moss. So it isn't, etymology doesn't really point you in the right direction. It just gives you some idea of how tortured our past is when it comes to trying to form words and ideas. So the real fungus is the underground mass of root-like structures called hypha. And that's why it was so long a period of time before it was realized that they were not part of the plant kingdom, which is where they were. They were in the phylum Thallophyta, hidden life it was called, I think was the name of it. But things they couldn't really nail down too well until about the 18th century and really the early 19th century when this first started to become clear. Um, and I make the point here, we're going to talk about edibility a little bit. They are made of chitin. They're not made of cellulose. They're different material, which doesn't surprise anybody. So that's what, and you can see why I took this picture out. It shows how the hypha really mash together to make the fruiting body come out of the ground, still attached to the thing at the bottom, which is really the fungus, this thing called the mycelium. And it is the fungus in every case. And some things that don't ever come out of the ground don't make a fruiting body. And we were talking about truffles before we started. Truffles don't create an above ground fruiting body, but they are, in fact, the fruiting body of the truffle's fungus, mycelium. So the mushroom features, just so you don't remember, we're talking about jelly fungi, which we'll get to shortly. But the mushroom is really at the heart of the fungal understanding and your interest because it's the only really macroscopic, interesting thing that is variable enough and interesting enough and edible enough and poisonous enough to elicit a fairly wide range of interest among many people. And so the parts are fairly innocuous. The cap, or it's really called a pileus in the language, but it doesn't really matter. This is called the stipe. The fascinating feature to me is this little skirt called an annulus around the middle for some fungi, and that is married in others to a universal veil or sac at the bottom, and those are to protect the gills underneath the cap, or pores. In this case, these are all gilled. Sometimes they can have pores. You can see this one is a pored mushroom. It's hard to see in that, but it doesn't matter. So the whole point I'm getting this is that the entire function of this thing is to get the spores out of the ground far enough away so they can be launched into space so that they can then propagate the species. And as you saw from the Ganoderma applanatum, the number of spores created is in the trillions for these large fruiting bodies. Mushrooms don't have near that many, but they have a lot, which is going to lead us to why mushrooms have gills or pores and why they have this exotic mechanism by which they protect them. That's what the veil is for, and the universal veil. There's two of them, partial veil and universal. And these things on the top, these little spots up here, those are actually what are called veil fragments. So the veil actually covers the whole thing when it comes out of the ground. This is what it looks like when it comes out of the ground. And I cut this one open so you can see it's getting ready. Now, why it does that is because this is the real life cycle. We're getting into this because we're almost done with this part, because we're getting to how important this whole spore find it. So here are the two spores, two different types, and there are things called mating factors. I won't get into that here. But then when they are come together, they quote mate, which is again a euphemism for, sexual union in our way of thinking. And then as it grows and eats and absorbs and gets nutrients, it gradually runs out of food or for any other reason that it decides it needs to reproduce like everything else. It will form a primordium underground, and that will eventually mature to the point that it's ready to come out when inspired by the proper environmental conditions. And then the spores are produced and the cycle continues. So this is what's going on, and we're going to get to taxonomy very shortly. The phylum is called the Dikaryotes, and you'll see that at the bottom here, Dikaryotomycota, and that means double nucleus. And the reason that is like that, and I like to make sure people realize this is really unique among fungi, how they propagate. These are the two spores we saw earlier that are compatible, and they find each other as wandering hypha through the soil. And it's discovered through some means that I don't know the distinction. It's probably not well known in the scientific literature either, how and when this is done, some sort of a signal. And then they start growing, and you'll see that what happens is there's the two different nuclei, and then they undergo mitosis, so they split. This isn't meiosis. This is just mitosis, just regular growth. And then rather than keep going that way, they'll actually form this little bridge that moves one of the green ones and one of the orange ones back to the next cell. And so it'll go through at the very bottom, it'll go through as N plus N. So it'll go through as two different nuclei, or dinucleotide. So when it gets time to reproduce, the dikaryon then it's time to make a mushroom, if you will, and that's here we are in the mushroom that's already up. That is where the meiosis occurs, and you get the actual formation of the spores, which have the full set of chromosomes. We go through life as 2N, right? 23andMe. So that it's only the sperms and eggs have 23 chromosomes. Every other cell in your body has 46. I think every other one, but people keep finding weird things going on. So I'm not going to say that's. I very rarely use always because it seems like that could never be true. But I point out that often enough is good enough for always in some cases. I'm an engineer, not a scientist, so therefore close enough is usually okay. You don't have to be perfect and exact. So because you got to get somewhere at the end. And here's the key distinction, and this will come up towards the end because here's the basidiomycetes with their 4 club-shaped mushrooms. That's called the basidia, this little structure. You can see it here. And at the very end of it, there's this little piece that comes out that holds the spore. I'm pointing this out because when we get to jelly fungi, which we're getting ready to dive into, that's really the only continuity that's going on here is that particular part of it. In the ascomycetes, which means cup or sac, it's a little bit different, but basically you get 8 spores. But that's the basis for the entire taxonomy at the high level. So we're sort of done with the part 1 into now the importance of spore production and dispersal as a key and really primary taxonomic factor. So Agricalus, that's the order of gill mushrooms. They have a complicated mechanism. We talked about how they protected the spores. And I point this, I think every, and I'm going to talk about briefly coral, tooth, puffball, stinkhorns, and then we'll go to jelly because they all have really bizarre ways of getting the spores out there. And that's how in almost every field guide, you will also find a separate section on coral fungi, on tooth fungi, on puffballs, and stinkhorns. They don't fit into anything because they don't do the same thing the same way, and they're all evolutionary, at least related some way, but I don't think we're there yet on all the exactitude of how that might fit together all that well. So why is a mushroom shaped like a mushroom? And there's a couple key points here that I talked about the primordium. It sits there underground waiting for the right moment. And I think most of you recognize that right moment is after a rain. Now, the reason that is because the mushroom uses the evaporated water that collects under the cap once the cap is open to enable it to shoot the spores away from the gills. Remember, the gills are hanging vertical. and they're right next to each other. And that is done to maximize the surface area or to maximize the amount of spores you can produce. If it was a flat surface, you'd have almost none. But if you have these eaves, then every eave is covered on both sides. So you've really maximized, but you need to figure out a way to shoot the spore away because it can't go down. It's got to go out and then down. This is how it does it. It actually captures a droplet of water. That water spreads. It's called the surface tension catapult and shoots the spore away. Now that's a really, really exotic method that evolved for this to happen. And in fact, in my view, this is probably the end point. And these other experiments, among which are the jelly fungi, went a certain distance but couldn't go any further because they couldn't get beyond a much more complex and limited and not as effective means of spore dispersal. So you don't see as many of them because they don't go as far, get as far, and spread as far. That's hypothesis. That's not factual in terms of, like many things are. You can't prove these things without experiments. So just a brief slide on the others in case you're not familiar with them. These are coral fungi. Usually you find one edible among, and by the way, these are mostly edible, although I don't think anybody eats them. And they're shaped like coral, and they're shaped like coral for the same reason that coral are shaped like coral, right? Coral are growing on a reef, and they are corals because they want to maximize their surface area. So like a tree, they branch, and that's why coral fungi branch so that they maximize the number of places where you can have spores produced. The surface of the fungus on which the spores are produced is called the hymenium. So you're trying to spread out the hymenium with as much surface area as you possibly can. So that's what coral fungi do. The most common one is this crown-tipped coral. And that's the edible one. It's called this cauliflower mushroom, which it is a coral fungus, but it's kind of deceptive, as are many fungi, by the way. So the opposite of that, so that's sticking up. Sticking down are the tooth fungi. Somebody mentioned this one. This is the lion's mane. It's got all kinds of other names: bearded hedgehog, satyr's beard. It's got a weird name in Japanese because it's some sort of a mystical warrior that from the woods. I don't remember what it is that seems to symbol them. But the Japanese are very, metaphoric about nature in many ways, and mushroom identification is a good example of that. Teresium erinaceus. That's why that one is still, as far as I know, still valid. And by the way, if you ever see a pan in my pictures, that's my frying pan, and that means I've cooked them and eaten them. So if you look, I'll do a short thing on the edible fungi. Short puffballs, a giant puffball in the upper right, cooking in the lower right as a sort of a breaded, sort of a French toast puffball arrangement. They're all basically the same. They're gleba. They're actually a separate group called gastromycetes, but they all are involved. They're small ones on the left. They all form a little hole in the top, and the pores actually puff out, which is why they're called puffballs. Interesting species. And finally, stinkhorns, which I'm sure you've seen or haven't, if you can imagine. And they, of course, don't produce spores in the same way. They don't put them in the air at all. They smell, and the insects come and fly into them. That's what the smell is all about, and fly off, covered with, spore stuff. All right, taxonomy. So here's here's the two kingdoms. I won't get into the Chromista protista thing, but protists is and the reason why I even bring it up here, slime molds are protists. So that's why we're going to talk about it very briefly at the end. But the kingdom fungi is here with the three phylums. The phylum Diceromycota, that is the one I talked to, and then the Basidiomycotina, and there are various names. I won't go into this. The different texts are still playing with the actual names. So you see Eumycota there. That's in some text, but they're still settling, and may never, by the way. But you'll see mushrooms and jelly fungi are all in that same category. So now we're going to focus from here on out on jelly fungi and jelly-like other masses that have similar characteristics. So that's my definition. I made that up based on my research, that it's a primitive form of agaric based on the evolution of basidia structure and spore release. That seems to be the only feature that is more or less consistent among the jelly fungi. So this is a kind of a rough estimate in the current research of what the phylogeny is for jelly fungi. And you'll notice we're under the phylum, right here, Basidiomycota. Then there are two subphyla that are also include separate at a very high level now, rusts and smuts. Now, you may wonder what rusts and smuts are. So you may have seen this, and there are many rusts. Wheat rust. This is cedar apple rust. I took this picture, by the way, over at what's the Sugarloaf Mountain at the base. It grows on red cedar, but it's a fungus. And then corn smut is a very famous delicacy, huitlacoche from Mexico, and that's it. I only point that out because there are lots of these too, but they're very high level forms of fungi. Back to this, yeah, I can't remember. Okay, so that's what they are. Now we go down into the jelly fungi. So there are three classes. Now remember, it goes kingdom, phylum, class, order, family, genus, species. So we're going down through now class. So the agricomycetes and tremolomycetes and dacrymycetes, those two that are bolded are jelly fungi. And then you go down one more level and you get to the orders. Under the agricomycetes, you've got all the guild mushrooms, puffballs, all the other orders are under there. And then separately from them, you've got the orders Auricularius. You may remember that from the tree ear. And this other one called Sebacenalis, which is, I don't have any examples of because it's fairly obscure around here. That's the only thing you're going to see with regard to. So what distinguishes these things is they have this basidia, this structure. Many, but not all, are septate, which means they have a little barrier along the length of the basidia. I don't know that there's any distinction that makes this unique, other than it seems to be an evolutionary process of getting to the basidium without any septa, which is what the agarics, I'll use that term, it's a little more easy to use, for the mushroom type, they don't have any septa. So the basidia are the structures that produce the spores, four spores. At the very end of them, there's this thing called a sterigma, and that's actually the place where the spore is attached and grows. So they're called hetero because of these septa. All others are called homobasidiomycetes. In other words, they don't have any septa. So I suspect that in the evolutionary progress, the septa were found not to be very effective, and so they got rid of them. I mean, by, again, random mutation. But they extend, the basidia extend through the jelly from the hymenium, where the spore surface where it grows, out into the air through the jelly, and then they release the spores. Sometimes it's a one spore creating another spore, a second spore, which is all really, really unique. Why is there jelly? I think it has something to do with the same function of the water for the guild mushrooms, only the jelly is also, obviously, it dries out, it gets water, so it retains water. So I suspect it's a similar mechanism that's doing the same thing using this particular technique, which is not as effective because they're subject to the sunlight and all kinds of other environmental factors that tend to make them rather transient. So here are three very typical forms. This is out of a book called The Fifth Kingdom by Bryce Kindred, and it's in my references at the end of the talk here as one of my, I would call it the Bible of practical mycology, perhaps, for me. So you'll see tremella. You'll recognize that maybe. That's the most common. They used to all be called tremella, and many of them still are. So it's called cruciately or four-way septate. Then you've got this dacrymyces. Remember from the dacry, we talked about that earlier. That's the one that's also up there as a class. They are forked, and they don't have septa. And then you have one that is transversely septate, and that's the auricularia. So they're all different in taxonomy, in this particular very detailed part of how they reproduce. So here's a more detailed drawing, and you'll see here it's a kind of a cutaway of this is the tremella. Again, that was the four septate with the four-walled septa shown here now. This is the jelly, the gelatinous matrix, all the way through, so it's embedded in this stuff. And then they stick out through the top, and that's where the spores form. With a similar basidio structure, and a stegmate at the end. The hymeneum is at the bottom, so they grow from down here. And these are the nuclei. So there are four spores for tremella. So here is a tremella, relatively common, but not as common as the tree ear. This one's called Tremella foliacea. I don't think they've changed the name yet. Jelly leaf, which really is what that means in Latin, by the way. Folia is leaf and tremella is jelly or gelatin. That's what it looks like. You can see that's about the right size. They're 1 to 8. This one was about probably almost 8 inches across and about 2 inches high. Like most fungi, they mostly come out in the fall for the same reason that fruit comes out in the fall, right? If they come out in the fall so the spores can get out into the air and then overwinter, and they're ready for spring to start growing through the soil as a hypha and then meet up with a partner and start creating a new mycelium. So that's all that's going. These are throughout North America on dead wood, stumps and logs, mostly just both deciduous and conifers. And as we will see, like most of them, although I don't know anybody that eats them other than the tree ear, they are all edible. I'm not sure how good a database we have on that, but and again, I don't know of anybody who's gotten sickened by eating one. So here's the Dacromyces, the one that didn't have any septa. And you can see it's called the tuning fork basidium. Again, here's the gelatinous matrix. Here's the hymeneum. So, and they only produce two spores, but they're jelly fungi. So they're obviously some other mechanism that's a different history that use the gelatin method to protect the hymeneum and keep the water close to the spores where they are then able to use that, probably the same mechanism. I don't think anybody's done any work on that. So here's a very common, one that you see fairly common, Dacromyces palmatus, called orange jelly commonly. Again, made in November, no surprise there. These are a little smaller, a little shorter. They can get almost red on only conifers. I've only seen them on conifers, and this is important because witch's butter, which looks a lot like this, grows only on deciduous trees. Only it's also mostly brighter yellow. You'll see that. I have a picture of it a little later. Again, edible. Nothing surprising there. You'd think the name orange jelly would give it away, right? Maybe that's how it got the name. I don't know. I don't think people smear these on toast, but they might. And finally, auricularia, and this is our famous tree ear. And here you'll notice they're transversely septate, growing through a gelatinous matrix and connected to the hymeneum. And again, once it gets out of the matrix, right above the matrix, of all of my looking at these things, I've never seen anything. Now that I know this a little better, I'll look next time very carefully to see if you can actually see anything sticking out of the top of the jelly. I suspect in certain cases you might be able to see something, but I'm going to have my eye out from now on. And of course, here's the tree ear growing in the wild. They grow prolifically. They're very common here. In Patapsco State Park, near where actually the Middle Patuxent Environmental Area, sorry. They are really all over the place. The whole tree trunks are covered with them, at least on occasion. Again, 1 to 6 inches wide, and they're much more wide almost through the whole season. And they are a noted edible. In fact, we're going to branch off now briefly into edibility because this is one of the world's most and oldest cultivated fungi. So it was first cultivated, according to the records, in the year 600 in China. Now you'll know, of course, that many fungi have a rather more developed history in both the Far East and in our own European origins in some cases. So this, you may have heard of John Gerard's Herbal. He's a very well-known 17th century herbalist, wrote about medicinal plants. And this is a quote. I just capture the time of the moment in the actual words. The fungus excrescence of the elder is a type of tree, I think you probably knew that, where the elderberry comes from it, commonly called Jew's ear, is much used against the inflammations and all other soreness of the throat, being boiled in milk, steeped in beer, vinegar, and any other convenient liquor. This is the therapy of the 17th century. And by the way, this whole thing about, this is Judas Iscariot, of course, the pieces of silver, and all the ways nature has tortured him. There's trees named after him, and the ones he was hung from, and there's interesting. So you see a lot of that in the older literature, in etymology of various phrases. And this is some more recent research. Just I point this out, and yeah, I have a whole talk on medicinals, but this is some of the things they found with some scientific experiments, usually in mice, that it reduced activity of solid tumors, and then a significant of lowering plasma glucose. Now, this one has an interesting, also modern thing. You may have heard of the it's called the Chinese restaurant syndrome or the Szechuan. Some people go and they eat these things in soup, and they give a real. What they do is they cause the platelets to decrease in number, so you get what's called purpurea. So you get these red blotches all over yourself, and so and people, it's a natural reaction. Some people have more than others, but it is attributed to this particular fungus, not MSG, by the way, which is another finger pointer that people get from eating in Asian cuisine on occasion. So fungi as food, not much on this, just to let you know that it's good to eat them, and people do. They have a lot of protein relative to plants, and they have about as more protein than any other food other than meat. They're very nutritious. Uh, some cautionary notes: a few are deadly, and you should never go out on your own without being with a great deal of confidence. But there are among the wild versions of edible fungi, there are 12 that I find that you could probably safely do on your own without too much. And they are listed here. You'll see tree ears number 4. And what that means is there's nothing that looks like this that is poisonous, with the possible exception of meadow mushroom, but you know that one as the white or button mushroom, which is the one we get at the supermarket. So, and you'll see some of the ones we've talked about. The bearded hedgehog is here, lion's mane, chicken of the woods, hen of the woods, chanterelles, morels. Most of these you've heard of. So the importance of mushroom production, the one that's circled, 18% of the world's cultivated mushrooms are tree ear. And so the mushroom industry is now pretty huge. Right here on the right is the wood ear production in China and how it's grown over the last 20 years. So this is really an important aspect of modern life. So, jelly, fungi examples. Okay, so here's Tremella mesenterica, witch's butter. As you can see, it's like the orange, only it's lighter. Again, year round, all North America. I see this most. I don't see it down here. I find these mostly up above about 1,000 feet. I'm sure you could find them, and I have, but they're much more prolific up in the Shenandoah Park area. That's where I find them mostly. That's where this was taken, and they're edible. Again, deciduous wood. The orange one grows on conifers. That's one of the reasons you can tell the difference. So this is a globally recognized, and you'll probably recognize the term witch's butter. Yeah, also yellow brain fungus, tripe fungus. By the way, mesenterica, the species, means middle intestine. So it's called the tripe fungus in English, among other things. And you'll see some of the other names from different languages. So it's pretty universal. Again, here's some medicinal benefits. It stimulates the immune system. And here's some research from Israeli and Ukrainian researchers from the year 2000 that had anti-tumor properties, and many fungi do. Uh, in fact, I don't know how many have not been tested for it, but for those that have been, seem to have been successful. So here's another one that is not uncommon. It's called white coral jelly. It looks a lot like the coral fungi in a way, but not really. It's also called dead man's rubber glove because it can take up sort of cylindrical finger-like, appearances on occasion. It's specifically Central Appalachia, but probably elsewhere. I think this was taken on White Oak Canyon Trail in Shenandoah Park. Summer and fall, you can see it's very, very spread out, gregarious, and you'll cover the whole log in some cases. And sometimes it has finger-like blunt tips, which is why it gets the dead man's rubber glove. And here is one of the more, I would say, lugubrious, it's called black jelly roll. You see this on occasion. It's also called black witch's butter. So because it has a similar characteristic, and it's also rose on deciduous wood, like witch's butter, only it's black. Also, the witch's name, you could think of black as probably more characteristic. In fact, I think in England they call the yellow one witch's butter also because of that. I don't know that, but there's a difference of having to do with the name witch that has something. And this one, I don't know anybody's eaten this one. This one's not listed as unknown. So you could be the first one to try it if you ever run across it. Okay, so that's it. That's it for jelly fungi per se. Uh, who they are, what they are, why they are, and what distinguishes them in the family of life in the kingdom fungi. So there are other things out there that you may mistake for jelly fungi because they can be sort of jelly-like depending on what it is you're looking for. And so the first group are the cup fungi, which in that other phylum, which we talked about, the Mycota, these are the cup fungi, the ASCII. They grow on a flat surface or curved surface. A cup is a curved surface, obviously. And it has something to do with water again, capturing the water in a cup. Then when the water evaporates, that is one of the mechanisms that is used by the cup fungus to spread its spores, which are spread out. The hymenium is the cup surface. So it doesn't have gills. It's got cups. And the water evaporation has something to do with it. Lots of stuff. There are actually many, many more ascomycetes than there are basidiomycetes. The largest phylum by far. Penicillin, yeast, aspergillus, cup fungi, and morels, of course. And all the algae associated with lichens are also ascomycetes. So here are some that you might see depending on how old and how beat up they get. This one is called the hairy rubber cup, and there's another one called the common brown cup that it doesn't have such a circular cup shape. It can look a lot like that jelly ear, and in fact, it also looks a lot like tree ear because they're all brown and they're curled up. So you notice the strange names like hairy rubber cup, shaggy scarlet cup. So this gets to the common name problem with fungi, and here are two more. Somebody mentioned dead man's fingers. So there they are. They're ascomycetes. And orange earth tongue. The reason why these bizarre names occurred because the first field guide, which was the Audubon field guide, didn't have common names when they issued it in 1982, and the editor said they got to have common names. So Gary Linkoff went out with some friends and made up all these things. And so they don't go beyond 1980. So the older ones do, hen of the woods, chicken of the woods, but no, none of these other ones, and they're weird, like most mycologists tend to be. This is a weird one. I ran across this. It's called deer vomit, slime flux. It's actually an ascomycete. You may have seen this. Every once in a while, it's pretty prolific. What it is, it's a fungus that grows on the sap. And I couldn't figure out for a long time what it was. It's probably a saprophyte. But again, this is research that's just really within the last I'd say 5 years. It's a really odd looking thing. And it'll cover the whole side of the tree sometimes. All right, finally. Slime molds and protista. So the fungi are some kind called the fifth kingdom because obviously there were plants and animals all the way through back to Aristotle and beyond. People recognize that distinction. But it wasn't until von Leeuwenhoek came along with the microscope. He was actually, by the way, worked in the fabric industry and he wanted a way to see the better look at the fabric. That's why he came up with a microscope. Then started looking at pond water and discovered these tiny things. And that originally gave birth to the name protozoa. First life is what he thought it must be. And then gradually that emerged to become protista. So it's the firstness in thinking that these were the first things that were ever there. So they were actually discovered, and protozoa became monera or single life. So actually, fungi was the fifth kingdom in that breakdown. That's the reason why they're called that. Slime molds have traditionally been categorized as myxomycetes, and by the way, myxa is Greek for nasal slime. But typically you'll find them in field guides, and many people think they're fungi, and they kind of look a lot like fungi in some cases. But we talked earlier about protists. You may remember euglenids, euglenoids. They're moving things like paramecia that have chlorophyll. So it's like a traveling plant. There's some really weird stuff in there, only because we don't know where to put it. We being biologists, being humans, I guess, not me. And there are about 1,000 species of slime mold. I'm going to go through just two. Actually, I have two quick pictures, and then first of all, this is what the slime mold does. It's a spore that becomes either this like a small amoeba-like thing or the swarm cell, and they unite one way or another. Then they cross over into this thing called a plasmodium, and this is the actual fruiting body part that grows and starts to grow and pulsate and moves. And you can actually watch it moving, which is why it is such a bizarre thing that's caused a lot of issues. and here are two examples of can't cart, and there could be red. You could see this as maybe being um, potentially a jelly fungus. They tend to also get jelly-like as they get older. This is called carnival candy slime, deadwood, same time frame. This was called wolf's milk slime. It forms into a stuff called toothpaste slime sometimes because it actually looks like jelly. Not this particular picture, but the two most famous ones are what's called scrambled egg or dog vomit slime. And this is the one you see in your garden and your mulch quite frequently. It starts out orange, yellow, and then turns. This grows, and there have been numerous cases. The Blob, the movie, the first movie with Steve McQueen, was based on an incident involving scrambled egg slime in the 1950s that scared the whole community because it was growing. They thought it was from outer space. So that was really the origins of the story on which the Blob was based. And then more recently in Garland, Texas, there was another incident. You can find this in the news. AP, that's the AP story. And that's a quote. If it's something right out of an aftermath television movie, a mysterious encompassing ooze dubbed the Blob. That was from the newspapers. And oh, by the way, it's associated with superstition with witch's troll. Troll cat is called to spoil the milk. So there's a whole history of this one also. And finally, this is our last slide. Many-headed slime. This is the one that grows looking for food, and it can get very slimy. Here are two pictures I've taken over the years. On the lower left is this phenomenon you may have seen in the news where they took and put oat flakes in these locations that were the current train station depots around Tokyo over here. And you'll see this is based on the 4 days, 0%, 0.5, and the number of hours. Within 24 hours, it had found all of the locations of oat flakes, and that this network looks like the train network of Tokyo. In other words, they found the shortest distance between every station based on the engineering of the Tokyo metro system, which made the news. And there's been a lot more experimentation with this since then about it being able to find things. And so it surges and retracts in reaction to food and finds its way to the food. Apparently, it does that in a very efficient way by nature's methods of detection of chemical sensors. Has nothing to do with the brain. Well, we're not really sure what the brain does in that way, but we know it's probably not what we do. So here are the references. I mentioned, here's The Fifth Kingdom by Bryce Kendrick. This is the Audubon Field Guide, which I still use, even though it's dated. 50% wrong, but I know it well. This is a good Mushrooms of West Virginia by Bill Rudy, is more local and more usable. You probably run into this if you ever done any mushroom. Michael Crows, he's pretty good on field identification, I think. And this is my website, Hiker's Notebook, which I have a fairly substantial section on fungi. And here's what that looks like. And it's not just pictures; it's really a full article with all kinds of the etymology of everything too, which is something. Wikipedia started to do that. I started it actually before Wikipedia did, but then again, things pass on. AI is going to get rid of this on me pretty soon, I think, but that's okay. I'm still writing it. I wrote a book, as Bronwyn mentioned. This is it. It's available on Amazon. It was about $50. It's now down to about 30, 35, so it's not too bad. It's a good book. And also, if you're interested, Mycological Association of Washington is still very functional. I was the president for 6 years. You can attend things just like we're doing here with a mushroom lecture. I think it's the second Thursday. I remember the date. It's either second Tuesday or second Thursday every month. But mawdc.org is the website. So that is the end. There's a slime mold. That's the scrambled egg slope. There's a mushroom. There's that one is actually puffball cut up and for cooking, and that's a puffball called puffball in aspic. One of the weirder things. So with that, are there any questions?
All right. Thank you, William. You can unshare. I'm going to put a spot. Let me unshare you here.
Okay, yeah, you can do that better than I can.
Yeah. Okay. There we go. I'm going to put a spotlight on you.
Okay.
And then if you have questions, you can put them in the chat or raise your hand, and I can call on you, and you can ask William directly. We do have a question here, Mark. What are jelly's predators?
So, I've never seen anything eating a jelly fungus. I've seen many slugs, insects. There's even a fungus beetle. There are insects that, and if you pick up a russula, which is a very common type of regular mushroom, you'll see it's been obviously been chewed on. I have a picture of a squirrel eating a Brussels in my backyard. But I've never seen, nor do I know of, there's no literature of them being eaten by anything. Now, whether it's because they produce a chemical that is inimical or harmful or not preferred by insects, I don't know. They don't last all that long. So they typically are like it's called deliquescing. When a mushroom or a fungus is done distributing its spores, it sort of melts into this gooey mass. So that's what I've seen those, but I've never seen anything eating them. So I don't know the answer to that. As far as I know, nothing.
All right, good question. Jared has asked, what influences the color of each jelly fungi?
So the color of fungi is really an interesting and fascinating subject. I thought about it often. I wrote an article on what is color to nature, and for some things, it's everything. Flowers, obviously, they attract insects with the colors. I'm sure that fungi have some purpose for color. I have been totally unable to discover any reference to what color does for it. I don't think it's easy to find this out, except in the case of lichens. So lichens are colored in order to protect their algae, according to the literature that I've read. So in other words, there are green lichens, there are orange lichens, there are red lichens, and that color has to do with shielding the algae, which are on just below the surface, from the UV light. Um, since fungi do not rely on insects except for the stinkhorns, which attract by smell, I don't know of any other function that the colors have. It's not for spores. It's not for sexual attraction. So I suspect it's just a factor that is a byproduct of some other necessary physiological function. And remember, color is something we see three colors. Most mammals don't. They only see blue and green. So there's a whole unknown world of what color does, why color is, that there's not any good way to do too many experiments with anything other than like bulls and red capes. And you certainly can't do anything with fungi. So as far as I know, there's nothing that would attribute it to a particular color for a particular function.
Interesting. Mark is asking about what is the evolutionary history? How do we know the original date? When did these fungi come on? And I want to follow up with that because are the jelly fungi, were they first in terms of evolution and then the guild and other fungi come out of that? Were they or not?
So this is a really, as you probably, I think the first question is how do they first originate? So in the original timeline of biology, where do fungi fit in? I think that is that the first question, part of that. So no, it doesn't matter too much. All this is speculative, as all evolution is. DNA does provide a means of identifying uh, what's called monophylogenetic, which means you have a single ancestor. And you probably read how complicated the human story is, right? With Homo neanderthalis and Homo denisovans and all the other different species that are now no longer thought to be necessarily part of Homo sapiens. So we're still struggling with us. So there is some work on fungi. The jelly fungi research that I did present where we are, I said phylogeny so far, it does appear that they are part of the early evolutionary experimentation. And usually that's what, the whole kingdom phylum is based on phylogenetic history. But the phylogenies don't necessarily follow the traditional breakdown of how we've been defining things according to Linnaeus' system, the Linnaean system. So that's why it's so. But you can't usually get a definitive answer on timelines. The way they do it, by the way, is mutation rate. And there's been some observed numbers that you can use when you count the number of mutations, i.e., different DNA sequences, and the number correlates to how long it's been evolving. And so they use that as a really loose metric to try and lay out this timeline. I think it's probably premature to draw too many conclusions from the work that's been done on that so far. Other than things are obviously, if the DNA is different, they're not the same thing. And even if they look alike, and if the DNA is similar, they are similar. But how close they are in terms of branching and evolution is still in laboratory stages, with some work done in some groups that are focusing on a specific type of life. And fungi don't happen to be that common in that particular set of research, with all the other issues going on with DNA. I
William,
did
would
search for that, by the way. Go ahead.
the
Sorry.
fungi the jelly fungi that you shared with us, is that the complete repertoire that you would find in Maryland, or are there other ones?
So I've been hiking in the area for 30 years. Um, almost, several times a week, always out on trails and and always stopping and taking pictures of fungi. I have thousands and thousands of pictures. And uh, I can only go by my own personal observation and what I find in the field guides. And pretty much what you saw, I have others. There aren't too many, but there are some. Uh, but that's pretty much it in terms of the jelly fungi that I have run into. And I have not limited myself to any particular type of trail. Everything from 3,000-foot mountains in Shenandoah Park to along the Patapsco River Valley. So all I can say is that's what I have found. And I haven't found anything that would discredit that particular experience.
All right. Sarah asks, and you mentioned this earlier when you and I were talking, has DNA sequencing disrupted the taxonomy of fungi like it has for plants and everything else?
No, much more so. Um, Gary Linkoff, who is the author of the Field Guide to Audubon, he gave a talk for the Mycological Association about 10 years ago, where he laid out a chart showing things that were in one family across the top, and along the bottom, along the side, were the different actual DNA relationships, and every classification was across the board looked alike, but was a different thing. So there are cup fungi that aren't cup fungi. They look like cup fungi, but they're not. So it just really ripped apart the entire taxonomy of fungi, and it's still being put back together. I suspect they're getting ready. I think they're issuing shortly, may have, I don't know, a new Audubon field guide that has the new names in it. Um, but I'm not about to start over. So it's estimated that that book is about 50% wrong. So that's how disruptive DNA has been to taxonomy of fungi.
So Mark is asking, I guess, I think that it has to do with jellies. Are they found in deserts or high altitudes, undersea and hot steam vents, um, or or is that just fungi in general, Mark? I'm not sure what you're asking.
So, most of what we know is from our local. So generally, this is northern hemisphere temperate forests. That's what we're mostly talking about. Um, Aquatic is a whole other story. There are aquatic fungi. I don't think any would be jellies, because the whole thing about the fungus is to gather water. You wouldn't need to gather water if you're underwater. That's why, by the way, our bodies are 80% water, because our cell structure is basically pretending it's underwater, because that's how it originated. So, in terms of deserts and mountains, I've done relatively high mountain climbing, up to 18,000 feet, not 18, 14. I wasn't really looking for fungi as much as I do now. There are lichens, definitely, and lichen is a fungus. They're everywhere in the high mountains. And deserts, I've been to Death Valley. I didn't see anything growing there, so I did. But again, I would say, to answer your question, nothing obvious.
Okay. Alan has, when and why did you catch the fungi bug?
Okay, that's actually so. That's really an interesting question. I'm a mechanical engineer, ocean engineer, and several other degrees, but nothing to do with biology. I was in the military, is in the Navy submarine service, and I came back from my last assignment in Hawaii. I'd never been walked. I was in Scotland for a while, and I did walk around, but I came back here and started walking in 1993 and joined a hiking club, and I became fascinated by all these strange things, and no one knew anything about them. I asked people, and not a clue. So one of my hiking friends invited me to the French embassy in 2002, and I went, and they had a display sponsored by the Mycological Association of Washington. And by the way, for 4 or 5 years, I wandered around picking up mushrooms, trying to identify them, and I couldn't do it. And on one of my hikes, I had a young man, and I was sitting by the side of the stream with my hiking friend. We had a pile of mushrooms. He said, "Oh, that's a russula." We said, "How'd you know that?" We couldn't figure it out from the books. His father was a biologist at Penn State. So I gave him all my pictures that I'd taken for 5 years, and he took them to his father, and they came back with names on them. That's how it started. And then once I learned about the Mycological Association of Washington, I went to the first meeting near where I worked. I worked at Carter Rock. And so that was 2003. I got on the board, was on the board for 20 years, and became president in 2016. That's how it started.
All right, great start. Do we have any other questions for William tonight? I'm looking. Did anybody have their hands up? Any questions about anything fungi related, jellies?
Or nuclear energy for that matter.
or nuclear energy. Well, this has been wonderful, William. I'm very happy to have you back, and we'll get. Oh no, Preston has his hand up. Go ahead, Preston.
Sorry, I couldn't get my dad to do it for me. My question was I've only seen those jelly mushrooms that look like raisins, the brown ones. Uh, where are could I find any others if I looked? Um, like uh, like at Cromwell Valley Park or something, Gunpowder Lodge. Um, any other different uh jelly fungi there?
So you raise actually a different question. Recall what I talked about with the life cycle of the fungi. It grows underground as a mycelium in the wood or underground. You don't see it. When it's ready, when it needs to, it will produce a fruiting body. I have gone back to many places where I have found copious numbers of chanterelles, morels, and the second, third, fourth, fifth year I go back and there is nothing. So they
Hmm.
are so unpredictable. There is no way to go back to a location with any certainty and finding the same mushroom. And that applies to everything, including jelly fungi. So there's no way to point you in a direction. You really have to do what I did, basically, be out there wandering around and just stumble across it. I don't think there's any other good way to do it. That's why so few people do, because you got to spend a lot of time out in the woods. And sometimes you go all
Yeah,
day and find nothing. That's when you're
yeah,
looking. I'm not really looking. I'm always looking, but I don't go just to look for mushrooms usually.
yeah. Me too. Whenever I find a mushroom, I'm not looking for them.
That's
It's the
that's the answer.
um it's the same with finding these guys. You seem to find them when you're not looking. Um, but I think the mushroom that I see most often is turkey tails. And
Oh yes, that's because
sometimes
they are everywhere. Everywhere. You can go in any woods with any logs. You almost always find turkey or for false turkey tails. They look alike. But yes, there are about 5 or 6 different types of saprophytes, polypores that are very prolific. You can find them in any forest.
I um I had this North Spore kit. I grew golden oyster mushrooms and I ate them. Uh, Um, so the I have I like mushrooms. I like eating mushrooms.
Good. They're good for you.
And
I've never
make
eaten.
sure you know what kind you're eating, Preston.
Yeah,
Yeah,
it's in
I've
these are the
never.
presentation. And oyster mushrooms are on that list, by the way.
Oh, um, yeah, but I have found a puffball once. Only time I've ever seen one of those. I've never eaten any jelly fungi or anything like that. But I have seen really big turkey tail mushrooms, like a foot in diameter.
That's probably not a turkey tail. They don't grow that
Hmm.
big that I've ever seen. They could get maybe a couple inches, but a foot, that's something else. There are lots of polypores out there
Mm.
that can get big. There's one that can get huge, but I
I've
don't
heard of.
think there's ever been a foot wide turkey tail unless I've been on another planet somewhere. And I don't know. I've never seen one.
Mm. Well, I've heard some. They're not turkey tails. Some like red mushroom can grow as big as
There
dinner
are some
plates.
large, they're called Ganoderma lucidum. The shining mushroom, as it's the hemlock polypore, it's called. They can get quite large, maybe a foot, and they are bright red when they first come out.
Cool. Well, I am going to keep my eye out for more jelly fungi from now on.
That sounds like a good plan and a good legacy to the talk.
Great.
All
We have
right,
a couple more
thanks.
questions. Yeah, we have a couple more questions. We have, what do we know about Chernobyl fungus?
Uh, it's this is crosses to my nuclear power background. Um, yeah, and again, I won't go into that in any great detail, but the in general, the effect of radiation on life is not really all that well studied because it doesn't really have too many places where you can go do that. And Chernobyl affords an excellent example of what happens to not just, fungi and plants, but animals in the area where you have radiation fields that are up to the lethal level, which is the LD50 for radiation is 500 rem, and you got well above that in certain areas around Chernobyl. And from what I've read, and you can actually go fairly close now, because of course radiation and contamination decays over time. Um, but the results are no different, by the way, than if you were to take any life form and irradiate it artificially. Because by the radiation from it's just gamma rays. It's the same electromagnetic radiation we get from the sun. It's different energies, but it's the same thing. You're just irradiating it. You're not doing anything different from the Chernobyl radiation than from any other type of radiation. So I don't think it's had any appreciable effect other than random mutations, which may cause a more rapid evolution of different traits, and that's why it's cancerous. Because changing your DNA is not necessarily a good thing, but that's how evolution works. So I don't know of anything specific about fungi in Chernobyl. I've never really looked down that particular pipeline. My wife is from Ukraine and she was from Kharkov, so she was there when she was lived there when Chernobyl happened. So I have some insight into the actual living through it.
Well, we're going to have you come back to have a talk on your other knowledge base of the nuclear energy.
I'd be more than happy to.
A couple more questions have come up. Alan has said that he had some dog vomit slime on his lawn, stomped on it. It looked to be blood coming out of it. Does the slime harm the grass, or is there any benefit just to leaving it?
So you had Follico septico or scrambled egg or dog vomit slime. It's called both when it's brown and it had red like blood splotches on it. Is that what you said?
Yeah, it seemed to be there was some like inside. No, when I was stomping on it inside, there seemed to be like another like a red substance.
I've never seen
And I
anything like
got
that.
nervous that it was something like a life form versus some kind of a fungi.
I couldn't, it could be something from the substrate that it picked up. Weird things happen with colors, as we were talking earlier. There is a couple of different polypores that are grow on the ground at the bottom roots that produce little droplets of reddish stuff, but nothing like what you're describing.
So, do they harm the grass? Is there any benefit to
No,
just
no, they
leaving
don't.
it?
They don't hurt. Well, yeah, I've never seen. I don't think I've seen them on grass so much. Usually they're on mulch. They're consuming the mulch. Of course they're decomposing, but the grass is growing on decomposed soil anyway. It's not eating the grass. It's a saprophyte. It's not a parasite. I don't. So I don't think it harms the grass. I've never heard that.
Thank you.
All right. We have, if it's so difficult to ID and all the names are changing, should I bother to get a mushroom guide, fungi guide, and give it a shot anyway?
Well, I have a wonderful time with the guides I have. It's really, if you're a rock hunter or you're a bird hunter, then it's all about the find and the quest. It doesn't matter really whether you're accurate perfectly scientifically, because no one else is either, hardly. So I would not hold off and wait for the guide to get right, because I don't believe at this point it's ever going to get right. Things are changing too rapidly. We're going to have to go to online stuff where you don't have published books anymore at some point.
And Karen's saying, do any of the fungi or jellies and the jelly fungi glow under UV light? Can you go out and find them that way?
No, generally there's a very limited number of fungi that glow, and they're pretty well known. The jack-o'-lantern, the honey fungus, which is called foxfire. Um, in general, fungi don't glow, and there's a whole hypothesis. And by the way, the glowing is one specific chemical reaction, same one that lightning bugs use. It's just a chemical, luciferase. Some mushrooms produce it, or some fungi produce it. It is believed it might have something to do with attracting insects at night, but there's not any work that's been done to verify that's what it is. So I think at this point, it's just one of those things that it does, that it's part of some other physiological process that has nothing to do with producing light for you to attract you in the forest.
It's not going to stop us from going out to the forest with the UV lights and trying to find them.
Yeah, well, you can go on UV and find all kinds of stuff. I don't disagree. You can. That doesn't mean they're doing it for you, or you can go and I don't know that they produce enough light for you to go find them. Even jack-o'-lantern,
Yeah.
by the way. I've been out at night, and you can't see them at night. You have to get them in a dark garage and get your eyes used to it, and then maybe a very faint glow.
Mm-hmm. All right. Well, any other questions? I don't see any, but thank you, Willem. This has been a wonderful presentation. You've inspired us to go out and look for some new and interesting things and take pictures, and keep looking, everybody. Keep looking, Preston. You just got to get out there, keep your eyes open, roll some logs, and see what you find.
Okay, you're all very welcome. It was my pleasure.
Take care, everybody. We'll see you not next Thursday, but the weekend after that. We'll learn about the Chesapeake bolide and what happened 35 million years ago. Take care, everybody. Take care. Bye-bye.
Thank you.
Bye.